All right, hi. Happy Friday, Friday Junior once again. Today I want to finish up DNA replication. That will leave us with probably a day and a half worth of notes. So we should be fine for the test next Friday. It's only going to be on this one chapter, so it's going to be short. Won't be our normal, 50-pointer with tons and tons of essay questions or, our normal thing. I also in Classroom put up a link for a video. I'm going to show that video as well. I think I'll show it after I take the notes. What do you think? Before the notes, after the notes? It's 2 minutes. Before? You want to see it before? All right, let me do this. Let me go to the notes here. And just re-explain what we talked about before. Wow, this is slow. Yeah. All right. Wow. Okay, so yesterday we had started talking about how DNA makes a copy of itself. I'm going to review what we did yesterday, and then I'll show you the rest of the video. And then we'll do the rest of DNA replication. But I think it will help if we just kind of pull out the cobwebs before we go forward. So remember that DNA polymerase is, like a lot of enzymes, very specific, very particular in the orientation that it works on a molecule. So because of that, and because of the fact that we've got two strands of DNA, one going from 5 to 3, and the other one going from 3 to 5, one's flipped 180 degrees. That means the enzyme also has to be flipped 180 degrees, which also means that as you open up this strand, one side is going to work towards that replication fork there in the middle, while another will have to work away from it. What we're going to focus on right now, what we focused on yesterday, was this top strand. Now, I just want to emphasize that in this picture, it's the top strand. It doesn't have to be the top strand to be what we call leading or working towards the replication fork. It could be the lower strand as well. It just happens to be the top strand in this picture, meaning that whatever molecule is working towards that replication fork is always going to be the leading strand, whether it's the one that's on top or if it's the one that's on the bottom. Okay, it doesn't have to be on the top. Sometimes the animations show them the other way around. So remember that DNA polymerase also is a little bit off in its ability to attach to things on its own. It needs a helper. It needs a buddy. And so that's what that RNA primer is for. RNA primer is put on by primase. Primase is an enzyme that adds RNA nucleotides. So we're showing this in red here because it is RNA. It is not DNA. And we also have to keep in mind that RNA is not supposed to be on there. So when we're completely done with this process, there's going to be an enzyme that comes in here and it's going to take off that primer and it's going to replace it with DNA. Okay. But to begin with, You have to put it on there because DNA polymerase won't work on nothing. It has to have something to attach to. Harper. It is also DNA polymerase, yeah. And there's different DNA polymerases. I don't remember which one that is. It might be 3. You just need to know DNA polymerase. It's always attaching nucleotides in the 5 to 3 orientation, but it's matching up with this existing strand that's 3 to 5. Okay. So remember we had our helicase. Our helicase is breaking apart these hydrogen bonds between the A's and the C's and the T's and the G's. Wait, that's not right. Between the A's and T's and C's and G's. Did I say that right? Anyway, breaking the hydrogen bonds apart between the nitrogen bases. Then comes in these little single-strand binding proteins. That helps to keep the molecule open so it doesn't snap back together. What's not being shown here is those topoisomerases that would be farther upstream that would also keep that DNA molecule from twisting back and forth and potentially breaking off. That would be catastrophic. We wouldn't want that. And this thing's just going to work. Our DNA polymerase is going to add on our blue DNA nucleotides towards that replication fork. And as it expands, as it opens up more and more, it'll just keep on working towards that fork. Okay? That's what we've been looking at so far. That is this top piece here of this replication bubble. So this whole thing is a replication bubble. But we've just been focusing on one of the 4 segments here, this one. Okay. That's all we've been looking at so far. What's that? Okay. So now I will show you that video. Lucas.
So the picture on the very top, that's like saying it like it splits in different directions. It starts in the middle and it goes in
right. So this guy here is going to start at the very
that's where like initially breaks.
Yeah.
And then there's also the one on the bottom is also.
And the bottom one's going to have to
strand.
That's to work back this way.
But in the other direction, it's happening at the same time going.
Yeah, going in the other direction too. I haven't gotten that there yet. Yeah. And if you really want to blow your mind, and I'll come back to this if it doesn't make sense to you, it's leading here, right?
Yeah.
But look what it's doing here. Here's the replication fork. What's it doing? It's lagging. It's going away from the replication fork. So it's leading in one direction, and it's lagging in the other.
But they're both going the same direction.
But is it 2 different things, or is it?
Yeah, they're both going 5 to 3.
Are they separate?
What's that?
Are they separate, like primers?
Yeah.
Or like,
Yeah, separate primers, different enzymes, the whole.
but they're on the same line.
Yeah. But you could have, many of these on one piece of DNA, opening up all simultaneously.
Okay.
So it might only have to go to over here, and then there's another bubble that would be meeting it going backwards.
Okay.
Yeah.
Just like a circle up to.
No, it's just straight. So this would be straight.
Yeah, it's been chilling for
like the top goes with the top DNA and the bottom goes with the bottom.
Right.
Oh, so it just goes like back, but then you come back.
Well, no, they never come back.
They don't touch.
So this one's just going to keep going this way until it gets to the end. And this one's going to keep going this way until it gets to the very end.
And same with the bottom. They never touch.
Right. And this one's going to go this way until the end.
Or no, isn't that
And this
one
one's
going
going to
the
go
other way?
this way till the end.
Wait, what?
Wait, aren't they going the opposite? The bottom arrow?
Oh, wait, it's in there.
Yeah.
You're right. Actually, it's going to be this way, but it's like this.
Yeah.
And this one is going to be like this. Nope, I did that wrong too. This one's going this way. Nope, no. We've never done that before. Can you tell? Not as clever with that one. All right. So let's watch that video. Now that I've confuzzled you completely. That was on this. And I'll just turn up the volume. I think it'll be okay.
Is the major enzyme in the direction
So because the lagging strand has to wait for the helicase to unzip this and unwind it, it can only do so much, and it's working in the opposite direction. So it'll lay down what it can. It'll get to the end of that segment, and then it has to jump backwards and do it again. So it creates little fragments, and those little fragments are called Okazaki fragments. They're named after the scientist who discovered them initially, and his last name was Okazaki. That's where that came from.
polymerase.
Okay, so they're saying it's going to remove the RNA. So this is another DNA polymerase molecule. And these little green pieces, they can't stick around. They have to be gotten rid of. Plus, if I can go back just 2 seconds. There, that's perfect. You notice how there's a little gap there? First of all, I've got to get rid of that. So that DNA polymerase is going to come along, and it's going to replace these RNA nucleotides with DNA nucleotides. It's also, and it's not showing this, but it's also going to have to do that on the leading strand. But on the leading strand, it only does it once because you only have one primer, right? And it just keeps working towards the fork. This one's going to have a lot of priming events because it has to keep going backwards as this continues to unzip the DNA. The other thing that happens is now there's a gap here. Those are not bonded together. So the DNA polymerase, this particular DNA polymerase, will take out those RNAs and put DNAs in there. But what it won't do is it won't attach the two, the sugar and the phosphate that are next to each other. Of two different nucleotides, it won't put them together. So that little gap has to be gotten rid of. You have to bond those two together. And that's done with another enzyme that's called ligase. It's called DNA ligase. And I think that's in the video. Yeah, and for simplicity, we're not going to go any further in the video than that. Yeah. So if you decide to watch this video on your own, stop around 2 minutes and 10 seconds. Because once it gets into this is theoretical, and we're not even sure this happens. And it's not something that you will need to know for exams, and I think it's better if we just leave it right there. Okay, so that's both leading and lagging strands in there. Now, what I want to do is I'm going to go back in the notes and just run through that with you now that you've seen both strands. So far, in the notes, all we've talked about is the leading strand. We've only been looking at this particular area right here. That's the only thing that we've looked at. Now what we're going to do is we're going to look at the bottom strand here, which in this case is the lagging strand. And remember, it's going to have to work in the opposite direction. It's working this way. So if it's working in this way, it has to continually jump backwards to do that. And I think, yeah, so talked about that leading strand. Oh, nice. I love it when it doesn't get rid of my writing. That's amazing. Trying to figure out which will be faster because I bet it won't let me erase either. Oh, it will. Okay. How about if I clear the screen? Will let me do that? Yes. Okay. Okay. To elongate the other new strand, it's called the lagging strand. DNA polymerase has to work in the direction away from the replication fork. And because of that, you end up with these little chunks where you have many, many primers that are called Okazaki fragments. And they are going to be joined together by DNA ligase after the DNA polymerase has come along and replaced that primer. So this next picture, I think I have it broken down. Yeah. So let's look at it in sequence. Again, this is the bottom piece that we're looking at. And it's working. I'm just going to put RF here for replication fork. So it's working away from a replication fork, and it's working in this direction. So here it added on a little primer. Here it's adding on another little primer. And then it will, as it continues to open up, it'll add another RNA primer as well. Yeah. So you can see here in the video, or in this stilled animations, let's do blue here. This is number 1. This is number 2. So DNA polymerase, they're calling it Pol 3. You don't need to know that it's Pol 3. You can just know that it's Pol. Is going to add on, again, in that direction, it's going to add on new DNA nucleotides. But it will never actually attach to that RNA primer. Okay? So there's going to be a little gap there. Yeah, Max?
Why does it only make primer in certain spots? It's scattered around.
Well, I don't know if there's a time period that it takes for that DNA polymerase to jump backwards and get to the fork as it opens. I don't know if they actually know why. It does look random.
And then why doesn't it need to be all along?
Why does it need to be on the leading strand?
Just
okay. Because the DNA primer, it physically needs something to hold on to. Okay. And so if the RNA primer is there, then it can hold on to it, and then it can start and do its job. And once it starts, it's fine. Okay. So it doesn't need it all the time. It just needs it to start, that initiation part. So that's fine until it gets to the end of a fragment. And then when it gets to the end of the fragment, it's like, crap, I need another RNA primer. So it goes and finds the next RNA primer and attaches to that and starts working back in the direction that it needs to go. And then it ends up, okay, I'm done. And it has to keep jumping back. Does that make sense?
Yeah.
Cool. Yeah.
So I understand that, it needs to attach onto something like a primer
Mm-hmm.
go and continue on, but why can't it just attach onto the part that was already synthesized?
Because if it's jumping backwards.
Oh, wait, I'm confused. No, I understand that.
You get it? Yeah, because if it's working backwards, it could attach to it, but then it would force it to go the other direction, right? It would force it to go in this direction, right? You can't have it go that way. So yeah, you're right. It could, in theory, do that, but it would go the wrong way, and DNA polymerase can't do that. So it would have to attach and always work in the one direction. It's one-way enzyme. Yeah, good. Third sequence here is just kind of showing the same thing. Sorry, my computer's really lagging today for some reason. There we go. So now I have an Okazaki fragment. I know it's a fragment. It's really hard to see in this picture because this little piece here, these two things are not attached. So the DNA nucleotides that are next to this RNA primer, they're not attached to each other. So they're going to have to be put together. And I feel like that's the same pictures over and over again. Is it? No, it's not. Okay. Okay, so now I've got my Okazaki fragment, and here is my DNA ligase, and my DNA ligase. Actually, I don't know if that's DNA ligase or polymerase. Let me look in the next picture, see what it says. Okay, that was DNA polymerase. So DNA polymerase, first thing it's going to do is it's going to take off the primer. So notice how now, instead of being red like it was here, now all of a sudden it's blue. That's because you've replaced it with DNA. And then in the next one, you're going to put DNA ligase in there, and the DNA ligase is going to put those two sections of DNA molecules together now that primer's been taken off. Right? All right. So that's called the lagging strand because of the fact that it has to have all these primers put in there. It has to have all the DNA polymerases come back and remove many, many priming events, not just one. And then it has to put all those pieces together, with ligase. Now, the other thing that I was trying to tell some people earlier, and I don't know how good a job I did on it, was if we look at that picture back here, this guy. Replication is always going to occur in the it's going to attach in the 5 from the 5 to 3 prime direction. So the problem is, because this is a bubble, you have actually, how many replication forks do I have in this picture? I have 2. So because of that, this side, if it's going towards the replication fork, that particular side is going to be leading, right? But on the same strand, it's got to continue moving in that direction, but now it's moving away from the replication fork. And our definition of leading and lagging is, does it go towards the replication fork? And so because the direction is different in this second piece, yeah, it turns into a lagging. So that top piece is now a lagging strand. Yeah. Yeah, exactly. You need to look at the origin of replication. Is it going towards the replication fork, or is it going away from it?
So what's it cross? Also going with the origin of replication.
Yeah. I mean, at that point, it would stop. At that point, it would stop, and the RNA primer that's there would meet up with that new strand of DNA, and then you'd have the other DNA polymerase come in, take off the primer, and then put it together with DNA ligase.
So it goes does the backtracking, and then it just goes.
Yeah.
Only no, if it's leading, it doesn't come back.
Leading's not going to come back. No.
Only when it's lagging.
Right. And once the legging gets to the end, then it stops because it's like, okay, here's the other side, I'm done. And it doesn't do anything else. Right. All right. That's as far as I want to go with this because I think it's enough for you to know do this today. We will talk tomorrow, either virtually or here. about proofreading. That's easier to follow than this stuff is.